Module 4, Part 2. So, in this part, we are continuing our conversation about actin-binding proteins, and we are going to focus on the ones we didn't cover in the previous part. This includes the motor proteins, and in particular for actin, the motor proteins that we are going to consider is myosin. And then there are a series of proteins that are involved in stabilizing actin structures. These include the CAP-Z and tropomodulin. I want to add another one, titin, which is not an actin-binding protein, but a myosin-binding protein. And the other proteins involved in the organization of filaments, such as nebulin, which we'll consider in this part as well. In addition, we'll talk about two regulatory proteins, troponin and tropomyosin, found in muscle cells. So, let's start with the motor protein. The general definition of a motor protein, it is a protein that uses the energy of ATP hydrolysis to move along a particular filament, and in this case of actin. The actin-based motor proteins are known as myosin proteins, but you will all see that another part of the topic, there are microtubule-based motor proteins as well. So the most abundant type of myosin, and the ones that it was first discovered and probably one we understand the most about, is known as the myosin 2. So I'm going to use this structure of myosin 2 to introduce this topic, but we will also look at some other myosins as well. And I should point out that the genomic data and the bioinformatics studies suggest that there are approximately 20 classes of myosin proteins, but the best known myosin protein is myosin 2, with myosin 1 and myosin 5 also pretty well characterized. So here is a structure of the myosin 2, and sometimes also called muscle myosin, because it plays an important role in muscle contraction, which we'll talk about in a minute. It consists of two heavy chains, shown here in green, light and dark, and these heavy chains have these long rod domains called the tail domains, which are wrapped around each other. So they are coil structure of two alpha helices. That is what holds these two heavy subunits together. On the N-terminus of these two subunits, there is what is called a head domain or globular head domain. It is these head domains where you will find the actin binding sites. Myosin interacts with the actin through the head domains, and you will also find the ATP binding sites, and the ATPase activity is also involved with these heads. Also point out that there are this globular head domain meets this long tail domain. There is a short region known as a neck domain. It is an extremely flexible domain. The conformation of the neck domain is very sensitive to ATP binding, as you will see in a minute, and is really important for the function of myosins. Myosin 2 also has 4 light chains. These are shown here in black, and these are 4 small molecular weight proteins that are associated with the head domain and also helps regulate its function. Myosin 2 is quite large, and you can even see them under an EM as shown here. Here are some of the members of the myosin superfamily. We already considered myosin 2, which is in the middle. The first one is the myosin 1. Myosin 1 is made up of a single heavy chain and 3 light chains. You can see that the characteristic head, then the neck domain, and then the tail domain of the myosin 1. Myosin 5 is similar to myosin 2, that is made up of 2 heavy chains, as shown here. You can easily see that, a tail domain and a series of light chains. Many of the light chains that are associated with myosin 5 are calcium binding proteins such as calmodulin, and the association of the calcium is important in the function of these motor proteins. So let's look in general how a myosin protein moves along an actin filament. So in the upper left portion of this slide and in this diagram, we are only looking at one head. But if a myosin has multiple heads, both of the heads will do this. We'll look at an example in a minute. Here you have a globular head of the myosin, its neck and the tail region, and it's bound to some kind of cargo. So it could be the plasma membrane, it could be the membrane of a vesicle, if this particular myosin is responsible for moving vesicle along the actin, or it might be an organelle. It depends on the class of myosins. But in this case, there is just this rod here, and you can imagine it can be any kind of cargo. And you can see the myosin head interacting with the actin filament as shown here. So it has a particular conformation, as this is known as a rigor conformation. And in this particular diagram, the myosin head is not bound to ATP. And when myosin head is not bound to ATP, it will bind to the actin filament very tightly. So on the other hand, what happens upon binding ATP, that is shown here, you get a conformational change in the head such that it releases its hold onto the actin, as is shown here. And then the ATPase activity of the myosin head itself will hydrolyze ATP to ADP, an inorganic phosphate. It won't release those components, but when that happens, you get a second conformational change such that the myosin head will come down and bind the actin again. But in this case, it moves towards the plus end. Eventually, the inorganic phosphate will be released as shown here in the step 3, and the ADP will be released as shown in the step 4. And the myosin will return to its original position, which is known as a rigor position. And in doing so, the cargo actually moved related to the actin towards the plus end. From here to here, from here to here, from here to here. So there are 2 points that I want to make about this. The first, I want to reiterate that calcium is required. Many of the light chains that are associated with myosin are calcium binding proteins, and the binding of calcium is needed in order for this myosin to function, although that is not shown here on this diagram. The other point I want to make is that all of the myosin tested so far, all of them move towards the plus end of the actin except myosin 6. Myosin 6 has a chain of amino acid inserted into the protein part, making up the head domain, and the addition of these amino acids actually make this myosin work in reverse. So to date, only one myosin, myosin 6, has been shown to move towards the minus end of the actin, whereas all other myosins move towards the plus end. It is interesting because myosin 6 plays a role in endocytosis, and it binds to endosomal vesicle and moves them towards the center of the cell. What is interesting is that the way the actin filaments are arranged in the cell, their plus end is usually towards the plasma membrane, and the minus end is pointed more towards the center of the cytosol. So if myosin 6 is going to move endosome from the plasma membrane into the cell, it makes sense that it would move along the filament in the plus end to the minus end direction, and indeed it does that. So I just showed you a model of how myosin moves, and there is a prediction that can be made of this model. The distance traveled in one step, or the velocity, should be directly proportional to the neck length. If you think about this picture in the previous slide, you can imagine that the longer the neck is, farther the myosin can reach towards the plus end. So let's look at this experiment, which actually tested a number of mutant myosins that were generated in the lab. What distinguished these mutant myosins from the wild-type myosin was that the increasing longer neck were genetically engineered. That is shown right here in the bottom. The first myosin is the wild-type myosin, and you can have an increasing length as you move along the x-axis. And the velocities, or the rate at which these myosin move down the actin filament, was measured. As it turns out, there is a direct correlation between the neck length and the velocity, or the microns per second to neck length. So this hypothesis is supported by this data. So up until now, we have been talking about what one head does, but many of the myosins have more than one heavy chain, and many have two heads. An example is myosin 5. So for a long time, there was a standing question among cell biologists: how do these myosins that have two heads move along actin filaments? There are two possibilities. One possibility is what is known as hand over hand, where the myosin heavy chains or the globular head would take steps along the actin filament in the same way that we walk. So alternating leading heads. A single myosin head moves 72 nanometer in the hand over hand movement. It would be like walking or stepping stone, where you place each foot on every stone. The step size is 36 nanometer, but the total step or the distance one head travels 72 nanometer. The other possibility, you can think of it as an inchworm model, where the leading head would take a step and then the other head would shuffle behind it. One head would be the lead always. In this case, you would have a permanent leading head. It would be like taking steps to one stone, then bringing both feet together on the stone, then stepping to the next stone, and always leading with the same foot. The step size is 36 nanometer, and the head only moves 36 nanometer in this case per step. So this can be tested. If the hand over hand model is correct, then the prediction is that the overall step distance will be about 72 nanometers. This is based on what is known as the neck length of myosin 5. On the other hand, if the inchworm model is correct, then the prediction is that the step will be 36 nanometers. Well, as fluorescence microscopic technique improved and the possibility of the labeling the proteins with fluorescence molecules improved, the experiment could be done where you could actually measure the step length of myosin 5 along the filament. So what scientists did, they labeled one of the heads with a fluorescence label, and they tracked by fluorescence microscopy using a single molecule tracking and measured the step length. And this was highly sophisticated technique where you can use the super resolution microscopy at this resolution, the exact data. So the data is shown here. Along the x-axis is a time in the second. Along the y-axis is the position of myosin molecule in nanometers. And you can see that the average step distance is approximately 72 nanometers, which support the hand-over-hand model of myosin 5 movement. So it is thought that myosin with two heads, at least myosin 5, move along the actin by walking, similar to the way we walk. So we have learned something about the biochemistry of myosin. We learned something about how they move, the biomechanical properties. So now let's take a look at the cellular function associated with myosin. There are the classical myosins, which we just discussed, myosin 1, 2, and 5. They play an important role in connecting the actin cytoskeleton to the membrane, for example, along with the cortex of the cell. Here you can see that the myosin head is bound to the membrane, which you can imagine that this is a plasma membrane. This myosin also plays an important role in endocytosis and vesicle trafficking by moving vesicles that need to be moved towards the plus end. In this case, you can imagine that the membrane, this could be a membrane vesicle of an endocytic vesicle. We'll come back to this myosin 2 in a minute. The role of myosin 5 is mostly vesicle trafficking. It moves vesicles from the minus end to the plus end of the actin in cells and also positions organelles. So in this diagram here, you can see that myosin 5 tail is bound with an organelle such as a mitochondrion and moving it around in the cell using actin as a roadway. But I want to focus now on the rest part is the myosin 2, which plays a role which is important for the muscle contraction. What is interesting about myosin 2 is that it can aggregate to form thick filaments. What happens is that the tail region associate with each other to form these very thick fibers that you can actually see in skeletal muscle cells. The way they align themselves is that all these globular heads are either on its end or at this end, and the tail region interacting in the middle region, and this is known as the bare zone. So these filaments are very prominent in the muscle cell. So to understand how they function in skeletal muscle contraction, we have to look at the structure of the skeletal muscle and look at the molecular characteristics of a muscle cell. So here is a diagram of a muscle cell, and you can probably see this diagram in another biology class, such as anatomy and physiology classes. Skeletal muscle cells are made up of very long multinucleated cells known as myofibers or muscle fibers. A single muscle fiber is a single long multinucleated muscle cell. Now, if you look more closely, you will see that each of these myofibers contain bundles of actin and myosin 2, and these bundles are known as the myofibrils. And if you look very carefully and closely, you will see that this is a very interesting banding pattern of these structures known as sarcomeres. They are described as a I band, H zone, A bands, and the Z disc. We're going to talk about that. A single sarcomere runs from a Z disc to another Z disc, as shown here. And each of these myofibrils has thousands of these Z discs that are connected end to end. They are lined up end to end. So what exactly are these sarcomere? We are going to look at the molecular level, but the sarcomere are just basically one contractile unit which make up the myofibrils. Again, repeating, in one muscle cells, you have these bundles of myofibril, and each of these myofibril have a specific zone which is delineated by one Z disc and another Z disc, and they are end to end. And many of these altogether make up one myofibril, and this is the contractile unit in a muscle cell. So here is a diagram of a sarcomere showing the individual proteins. The Z disc is shown here in purple, and one sarcomere goes from one Z disc to another Z disc. So what you would find inside this sarcomere are actin filaments, which are shown in red. I want to point out the orientation of the actin filament. The plus end of the actin filament is interacting with the Z disc right here. The plus end is interacting with the Z disc. And the minus end is free here in this H zone right here. This arrangement is also true on other side of the minus end in the H zone, and the plus end interacting with the Z disc. So in the next one, this is also true. So the plus end is interacting with the Z disc, and another Z disc, another Z disc on this side, another Z disc, another Z disc on the other side. You also have these myosin 2 thick filaments that are found in between these actin filaments, which are shown here in blue. And you can see the globular heads at either end of the myosin thick filament interacting with the actin filament. Then in the Z disc, you find a series of proteins, filamentous proteins, that come together to form this Z disc. This structure is extremely stable. So unlike other cell types where the actin is very dynamic, it is very different from those. It is growing and shrinking all according to the cell's needs. Needs include the motility, vesicle trafficking, cell division. In muscle cells, this structure of actin and myosin together is extremely stable. So how do the skeletal muscles stabilize this structure? So the way in which they are stabilized is by actin binding proteins and the myosin binding proteins. Sometimes these are collectively called as a capping proteins. There are 4 very important actin or myosin binding proteins that stabilize the structure of the sarcomere. One of them is CAPZ, which is an actin binding protein that binds to the plus end of the actin filament. There is tropomodulin, which is another actin binding protein that binds to the minus end of the actin filament. There is nebulin, which is an actin binding protein that binds on the side of the actin filament. Now, one myosin binding protein in this group is titin, which binds to myosin as well as the Z disc proteins. The troponin or tropomyosin, on the other hand, work together and regulates the binding ability of the myosin to actin in a calcium-dependent manner. So let's look at how these 6 proteins contribute to the stability of the sarcomere structure. So here is tropomodulin and CAP-Z. As I mentioned that the CAP-Z binds to the plus end where the actin filament, as shown here in green, that's your CAP-Z, and the tropomodulin binds to the minus end, which is in the clear zone here. You can remember that from your first part of this topic that the tendency of the minus end to lose monomers. So you can imagine that if there was no tropomodulin here, the monomers would be lost. So the tropomodulin really prevents the depolymerization from occurring from the minus end, a very important function. So here is nebulin shown in this dark green line. Nebulin binds to the side of the actin filaments, which confers even more stability to the structure. When you have a titin, which is the myosin binding protein, and it is this spring-like or this curly Q-like protein in this diagram that is bound near the globular heads of the myosin, and it also interacts with the protein of the Z disc right here. Another 2 proteins responsible for myosin binding to actin are troponin and tropomyosin. Tropomyosin is a coiled coil dimer, lies along the muscle thin filament. Each tropomyosin dimer covers the actin monomers in an end-to-end fashion. The tropomyosin can remain in 2 forms. One form blocks the binding of myosin to actin, and the other forms open up the myosin binding sites on the actin, as shown here in the right. Now, this tropomyosin is regulated by a second protein, troponin, which is a calcium binding protein. Troponin has 3 peptide chains. These 3 peptide chains, T and C, binds to the calcium and controls the TNI and the TNT, which binds and control the movement of tropomyosin. So let's review the whole story. When a muscle cell receives stimulus from one of the neurons to contract, the stimuli is known as an action potential, release a large amount of calcium due to the action potential from the smooth endoplasmic reticulum of the muscle cell, which are extremely diversified and form large sacs and network throughout the cell known as sarcoplasmic reticulum. So I'm again repeating one time. When a cell receives a stimuli in the form of an action potential from a neuron, it triggers release of large amount of calcium from its sarcoplasmic reticulum, which is a modified smooth endoplasmic reticulum of the muscle cells. So the released calcium in the cytosol binds to the TNC portion of the troponin, which changes the conformation of the TNC and finally the TNI and the TNT. This conformation change moves the tropomyosin away from its original conformation that can occur, and you will see in a minute. Once there is no stimuli for the muscle contraction, the available calcium is transported back into the sarcoplasmic reticulum by calcium pumps, and the calcium concentration level of the cytosol drops, causing the troponin to lose calcium, and it again changes the conformation, and through the TNC or TNT, it changes the position of the tropomyosin as well, and it will be blocking the myosin binding site on the actin, preventing contraction. So the role of these 2 proteins is to facilitate muscle construction in presence of calcium and preventing in its absence. So basically, tropomyosin blocks or uncovers the myosin binding site, which is controlled by the troponin. So how do all these work together to mediate contraction in the skeletal muscle cells? So again, here is your sarcomere right here. Here is your sarcomere, and this is what it looks like in a relaxed position. So when you have a relaxed skeletal muscle, you have the actin filaments, you have the myosin thick filaments in the middle, you have the Z disc on either side, shown here in purple. So this is one sarcomere. When the muscle gets a signal to contract, and the signals are usually neuronal signals, as we talked before, ATP is hydrolyzed, and of course the calcium is required, as I mentioned that earlier, through the energy harnessed by ATP hydrolysis. All of these globular heads will move towards the plus end, from here towards the plus end. They will come off actin and step towards the plus end in the manner just I described. When that happens, because the whole structure is so stable and the two Z discs actually contract and come close together to each other. Now, this is just one sarcomere. This is just one sarcomere, but you can just imagine the hundreds of thousands of sarcomere are lined up end to end in a coordinated fashion, and this is what confers the contraction of the muscle cell and in the muscle. That is an important and well-characterized role of myosin 2, but I don't want you to get the impression that myosin 2 only functions in muscle cell. It has an important role in non-muscle cells that is summarized here in this slide. One of the roles of myosin 2 is to interact with the actin in the contractile ring of the dividing cell and to mediate cell division. The importance of myosin 2 in this function was shown in the following experiments. If you take a cell and artificially knock down the function of myosin 2, and there are a couple of ways to do this. You can delete the myosin 2 gene, or you can inject antibodies to myosin, and they will bind to the myosin and inhibit their function. Regardless, if you disable myosin 2 and you allow these cells to divide, what happens is that there is a failure of the cell to divide, and you will end up with one large cell with multiple nuclei in it. The nuclear division occurs, but the cell division cannot. Here you can see an untreated control. You allow several rounds of cell division. You get normal cell division, but in absence of myosin 2, the cell cannot complete cell division, resulting in one multinucleated cell. Another important role of myosin 2 is to interact with the actin in the region of the epithelial cell known as the adhesion belt, sometimes also known as the adherence ring. The adhesion belt is a band of actin and myosin 2 that is found right below the tight junction. Remember we said that the epithelial cells are polar. They have an apical region and they have a basolateral region, and in epithelium they interact with each other through these tight junctions. Right below the tight junction is the belt of actin and myosin 2. The role of myosin 2 is structured to actually confer stability to the cell-to-cell contact between the two epithelial cells. So it strengthens the cells-to-cell interaction in the epithelium. And during embryogenesis, the epithelium folds to form different structure, and it is the action of myosin 2 moving or traveling on the actin filament in the adhesion belt that facilitates that. And when you talk about the cell-to-cell interaction a little later in the course, we're going to look at this more specifically. Finally, in cells that display actin as a stress fiber. Here is an example of a fibroblast. You often find myosin 2 interacting with stress fibers, and this adds the mechanical stability of these stress fibers, and then of course this confers stability to the cell. So the next, here is a very short segment of this topic we're going to cover, which is intermediate filaments. So let me remind you that these are the rope-like filaments that is found in most cells. They are about 10 nanometers in diameter. They are so named because they are intermediate in size to actin and microtubules. They are rope-like polymers made up of intermediate filament proteins. Their main role is to provide structural integrity to the cell. They form extensive network within the cytoplasm, and although not shown here, you find some intermediate filaments interact with the cytoskeletal components such as microtubules. In cells, you also find intermediate filaments at the tight junctions, right here, as being shown here near the tight junctions, and an abundance of intermediate filaments surrounding the nucleus that is known as a nuclear lamina. It simply protects the nucleus from the shear force within the cell. So again, their main function is for structural integrity of organelles and of the cells as a whole. So what is interesting about the intermediate filament is that even though there are a lot of intermediate filament proteins that make up a lot of intermediate filaments, the monomers or the way those monomers are put together is almost always the same. And it is shown here in this second slide. So here in this intermediate monomer, it has an N terminus over here, and you have a C terminus over here. The first step is the assembly of an intermediate filament is to form this parallel dimer. It is known parallel because both the C termini are on the same end of the dimer, and both the N terminal are on the same side of the dimer. So then two of these parallel dimers come together to form an anti-parallel tetramer. In this case, it is known as anti-parallel because the N-terminal pair of one dimer is now closely associated with a C-terminal pair of the second dimer. A number of these anti-parallel tetramers come together end to end, shown here, and forms what is known as a protofilament. Four of these protofilament come together and they form a protofiber. And this protofibril, then four of these come together to form an intermediate filament. Again, what is interesting is that there is a lot of intermediate filament monomers, but they all come together in the same way to form an intermediate filament. So they are fairly large. You can see them on the EM, and here is an EM of an intermediate filament. So the most abundant type of intermediate filament are keratin and can be acidic or basic depending upon the amino acids in their polypeptide, and classified as type 1 acidic and a type 2 as basic. I'm sure that you have heard about keratin a lot of times. In large amount, they can be extremely rigid. Think about the horns, antlers, and hooves of the animals. They are modified epithelium, and the strength of these structures are provided by the large amount of keratin as well. On the other hand, in the humans, your nails and your hairs, they contain epithelial cells which are full of keratin as well. So they are very rigid. The type 3 protein includes the desmin found in the muscle cells. GFAP, or the glial fibrillary acidic protein, is found in the glial or supporting cell of the central nervous system, which includes the astrocytes and ependymal cells. And these astrocytes and the ependymal cells do have different function. Astrocyte is a star-like cell, supports the neuron. Ependymal cells produce cerebrospinal fluid, and particularly this is important during the developmental stage as well. The vimentin is found on the mesenchymal cells, which is the embryonic connective tissue cells from which all the connective tissue develops. The type 4 is a neurofilament, and the proteins are NFL, NFM, and NFH, which stands for neurofilament protein low, medium, high molecular weight. These intermediate filaments are responsible for the organization of the axon and maintain the axonal diameter, which is essential to maintain the cell integrity and axonal transport of neurotransmitter by motor proteins on microtubules. They themselves do not carry any cargo, but help in the organization so that others can carry the cargo that is needed. The last one is lamin, and it is found in the nucleus under the nuclear membrane, which forms the nuclear lamina. Although some of the lamins are found in other tissues as well. Please do not confuse lamin with the laminin protein, which is a connective tissue matrix protein found under the epithelial cells in the basal lamina. The chapter opener picture shows the green laminin protein in the basal lamina, which is not the lamin protein that make the intermediate filaments. So here is an immunofluorescence micrograph of the glial cells. Mainly these are the astrocytes. And there you can see that there is an abundance of these intermediate filaments in these glial cells. In this image, the nuclei are blue and the intermediate filament protein is stained green. And the monomers make up the glial filaments in this case. Is known as GFAP. You can see these intermediate filaments filling up the cytosol of these glial cells. So here is what those glial filaments look like under the EM. Now, all neural cells have intermediate filaments, but they are not always made up of the same monomeric protein as found in the glial cells. So here is another kind of intermediate protein found in the nerve cells, which is known as the neurofilaments. Even though they are put together the same way because their monomers are different, you can see that they have unusual branching pattern which you don't see in the glial filaments. So this all come together because of these monomers. So keratin is an abundant epithelial cells, especially in the skin cells. This is an immunofluorescence micrograph showing the keratin in an epithelial cells, and we are very familiar with the keratin, the presence of keratin in our nails, on our skin cells, and also in the cells which make up the hair. This concludes Module 4, Part 2.